Hello everyone and welcome to today's video. I am Dr. WD40 and today we are going to talk about the Hardy-Weinberg principle. This is a very important principle in genetics because it helps determine the frequencies of certain alleles in populations from one generation to the next. This is important because it can help determine the sort of pressures being put on a population if those frequencies aren't staying the same. So if Hardy-Weinberg principle or equation doesn't equal out, it suggests that the population is evolving. So let's get down into it now. So here, as a little review, hopefully you remember this by now. If we have a heterozygote allele right here, remember this individual has two different chromosomes. One chromosome carries the big A allele. Let's say we're looking at rabbits and we're looking at white versus black rabbits. For black pigment to be expressed in the hair, it has to have the dominant form here, which is the big A, and the recessive version then would be a white rabbit or little a, little a. So we can start looking at this in terms of populations. Now, there are a few different things that need to hold true for the Hardy-Weinberg to work properly. So there must be no mutations. So these are things where we're not looking at changes in the frequencies from one generation to the next. So no pressures at all. So no mutations. We don't want a random mutation coming in and making a different fur color. Another one is no genetic drift. So for no genetic drift, we're looking at larger populations. So it's not shifting some, okay, now we have 80% black rabbits in the next population. Only the white rabbits reproduce, so now we have 80% white rabbits. Or I mean, not population, the generation. So no genetic drift. So the conditions for everything to stay the same from one generation to the next. And also no gene flow. So no new genes coming into that population to change the dynamics. So not causing new selective pressures and so forth. And another part is we have to have random mating. So no sexual selection here. So you're not always picking the dominant form cross with the recessive form. It's random mating. There's no mate selection here. And then the other biggest one is no natural selection. So no selective pressures, so no environmental changes, no predators eating a certain rabbit fur color or so forth. So that's the biggest parts for Hardy-Weinberg. And if the Hardy-Weinberg equation doesn't hold true, it usually suggests something like this is happening to the population. So now let's start breaking this down. So it is the Hardy-Weinberg equation, so we're gonna need an equation out of this. So we're gonna write the alleles down again. So remember big A, or big A, little a, so the homozygous dominant or heterozygous, we have, so we're using rabbits as our example here, black rabbits, and little a, these are our white rabbits. So now let's make a random population of white and black rabbits. So here I'm just drawing all the possible ones we could have in this population, and I'm just making random ones. So this is our random population of rabbits right here. So draw a little dash around this. Then little spot, let's say, you know, in a somewhere in the woods, some woods, these are the rabbits that exist. So now from this, we can calculate the frequencies of each allele. Remember, individual alleles are separate in the organism. So let's say first we want our total population size. So total alleles. So right now we're deriving the equation. So total alleles, 2, 4, 6, 8, 10, 12, 14, 16. So 2 for each. Then we want to say how many of them have the big A. How many of these alleles are the dominant allele? So you can go through and count them. 1, 2, 3, 4, 5, 6, 7, 8, 9. So boom. we have 9 big A's. So now if we want to calculate the frequency of the dominant allele in the population, all you have to do is take 9 over the total alleles, 16, which is equal to 0.56. So 56% of the population has the big A allele, or is the big A allele. Then, how about little a? For this one, you can go through and count each one. Or, if you have 16, you can do 16 minus 9, which is equal to 7. And then same thing here, you do 7 over 16, which is equal to 0.44. Now's where the equation comes out. So for the dominant trait here, we use the term P. So it's a lowercase p for the dominant form, and for the lowercase form, or the recessive form, we use Q. So if we look at these results here, we can write the first part of the Hardy-Weinberg equation. P plus Q is equal to 1. So what this means is all the alleles in the population are 100%. So pretty straightforward. As straightforward as an equation can get, really. But of course, we're going to take it one step further. So the Hardy-Weinberg equation, what that takes into account is the squaring of each of these sides. So if you use your binomial expansion, if you remember that, we have p squared plus 2pq plus q squared is equal to 1. This is the Hardy-Weinberg equation. So, but let's derive that using what we know with Punnett squares. So what we've done so far in genetics is simple monohybrid crosses. So big A, little a cross big A, little a. You're looking at individuals in a population, but now we're looking at a population, entire population producing another generation. We can still express this using a Punnett square. So if we look at this, we can write it out. Let's say we are doing this cross. This cross is actually how we get to that derivation. So if you write this Punnett square, I'm not going to go over the specifics of making a monohybrid cross in this video, but if we draw this out, these are the chances of each one occurring. So big A, remember we calculated the frequency of that in the population. Now we can add that frequency into this Punnett square because we're looking at the chances of mating in the next generation, or what the frequencies of the allele should be in the generation. So this one then is 0.44, 0.56, 0.44. So if you were to fill out this Punnett square like you were before, so there's heterozygote, and little a homozygous recessive. So in this one, we can now put our percent chances of getting homozygous dominant in the generation. So here, this is equal to, remember, A is P times P, or P squared. So in this case, this is 0.56 squared, which is equal to roughly 0.31. This one then is equal to P for the big A times Q for little a. So P times Q then is 0.56 times 0.44, which is equal to 0.25. This one is also P times Q, so this one is 0.25 as well. And little a, this one is Q squared, or Q times Q, 0.44 squared, which is equal to 0.19. So I don't know if you see it yet, but the Hardy-Weinberg equation is hidden within the results in this Punnett square. So here we have P squared. Here is 1 PQ. Here is the other PQ, so that's 2 PQ. So P squared plus 2 PQ. So this represents the heterozygotes. This represents the homozygous dominant. And then we have Q squared, which represents homozygous recessive. That all equals 1. Now, does it actually equal 1? So 0.31 plus 2 times P times Q, or you could say each one is 0.25. So 0.25 here plus Q squared 0.19. So 0.25 plus 0.25 plus 0.19 plus 0.31. Guess what? Equals 1. So it works. And that's how this equation comes about. So last thing I want to talk about today is how we can do an example of this. These examples could be used a lot for monitoring human genetic diseases. From this equation, you can calculate something like this term right here. So if you are big A, little a, you're a carrier of a trait. So if you know the frequency of a certain disease in a population, you can then calculate the percent of the population that carries that particular disease. We're going to keep it a little simple now for this video, and I'm just going to show the rabbits, white and black rabbits. So let's say we have a population of 20 rabbits. 20 rabbits. Now, just making up numbers here, we have 15 black rabbits. And if we have 20 rabbits total, then we have to have 5 white rabbits. So remember, 15 black rabbits, that's big A, little a, or big A, big A. And then this one is homozygous recessive. Okay, now what do we want to find here? We want to find the following. We want to find P. We want to find Q. We want to find 2PQ. We want to find P squared. And we want to find Q squared. So where do we start with this? So the easiest spot to start here is the homozygous recessive. Why? Because you don't have two terms involved here. Remember, little a is Q, or little a is Q squared. So your Q squared is right here. So Q squared is the first thing you're going to calculate. So Q squared is equal to 5. So we have 5 white rabbits total over 20 in the population. Now it'll give us the frequency of white rabbits, or little a in this population, and this equals 0.25. So there, Q squared is equal to 0.25. Now, what's the next thing we can get? We can get Q. So if Q squared is equal to 0.25, and we take the square root of each side, Q is then equal to 0.5. Now, if we're looking at this, we can take this one step further, and now we can calculate P. So you remember this equation from earlier? P plus Q is equal to 1. So here, if we solve for P, we have Q. The P is equal to 1 minus 0.5. I guess you can see why I'm choosing the numbers I'm choosing. P is then 0.5. Now we have P and Q. Well, now that we have P, we can then square this. So P squared equals 0.5 squared, which is 0.25 again. And now finally we can do 2PQ, which is equal to 2 times P. And here, a lot of times people get confused and they use P squared or Q squared. So make sure you're choosing the right one here. So P is 0.5, Q is 0.5, and guess what? This magically equals 0.5. Real tricky. So 2PQ is 0.5. So if we look at it, we want to check ourselves. Always check to make sure it equals 1. Because then you know if you threw in accidentally threw in a Q squared here instead of Q, your answer won't equal 1. So here, P squared plus 2PQ plus Q squared, so that's 0.25 plus 0.5 plus 0.25. Guess what? We equal 1. So it checks out. So here, now you can see the total population frequency is just given this little piece of data. So I hope this video helped you understand Hardy-Weinberg a little bit better. And if you have any questions, feel free to leave a comment. All right, have a great day. Hello again. Thank you for watching my video. I'm glad you could come by. If you enjoyed this content, feel free to do that thing where you like and subscribe. Also, if you're interested, I prepare and record all of these videos live on Twitch. After the recordings, we do a post-recording question and answer section, and then sometimes I play games where any question is fair game as I game. Also, on Twitch, I'm a co-founder of a wonderful community called the Knowledge Fellowship, or TKF. Our goal is to seek and share knowledge with others while live streaming. We feature streamers who cover biology, law, physics, chemistry, geology, photography, health and fitness, psychology and mental health, rocket science and aeronautics, programming, and tons of different practical EDU streams. Feel free to check out TKF at the link in the description and join the very active Discord community. Hopefully, find something that interests you. With that, I'll see you all next time and have a wonderful day. And remember to seek knowledge and challenge everything.